Monsieur le secrétaire perpétuel, chers conférenciers invités, chers collègues, mesdames et messieurs, étant donné que les deux conférenciers invités à cette séance s'exprimeront en anglais, vous me permettrez, j'ose l'espérer, d'également présenter ce discours d'accueil en langue anglaise. Dear invited speakers, dear colleagues, ladies and gentlemen, the non-invasive imaging and quantification of insulin-producing cells in the pancreatic gland is considered since more than a decade as a high priority field of investigation by the National Institute of Diabetes and Digestive and Kidney Diseases of the US National Institutes of Health, by the Juvenile Diabetes Foundation International, and more recently by the European Commission. Already in June 2000, at the occasion of the annual meeting of the American Diabetes Association held in San Antonio, Texas, we presented a short communication entitled GLUT2-mediated transport of D-mannotulose, a tool for imaging of the endocrine pancreas, ? Such a proposal was based on our finding that D-mannotulose, a 7-carbon sugar extracted from avocado fruits, enters into cells at the intervention of the glucose transporter GLUT2, indeed expressed in insulin-producing cells. The same year, at the occasion of the Claude Bernard Lecture, delivered at the annual meeting of the European Association for the Study of Diabetes, held in Jerusalem, Israel, we reviewed our pilot work not only on the use of the nanoeptulose in the perspective of the non-invasive imaging of pancreatic insulin-producing cells, but also on a list of other possible candidates for the same aim, including 2,14-sialoxane, 6-desoxy-6-125 iodine D-glucose, 65 zinc, tritiated glubentamide, tritiated methiglinide, a 125 iodine-labeled mouse monoclonal antibody directed against pancreatic beta cell surface gangliosides, and the labeling of glycogen, which in situations of sustained hyperglycemia, for instance, in spontaneously diabetic animals, accumulates in large amounts in insulin-producing cells, but apparently not so in acinar pancreatic cells. The progress of our investigation on the use of D-mannotulose was hampered by the fact that research projects on this issue submitted to different granting institutions were turned down in the light of an objection, first formulated by one of my past pre-doctoral fellows, that this approach was considered not applicable to human subjects because GLUT2 would be expressed at very low levels and functionally not essential in human beta cells. Yet, as underlined in the review article that we were invited to contribute for Nature Reviews Endocrinology, we indicated that already in 1966, D-mannotulose, which inhibits glucose phosphorylation in islet cells, and by doing so suppresses glucose-stimulated insulin release, was reported to inhibit insulin secretion in human subjects, this being confirmed in 6 further studies published between 1978 and 1972. Moreover, already in 2001, we indicated that in isolated human pancreatic islets, D-mannose accumulates intracellularly, inhibits glucose catabolism, and suppresses glucose-stimulated insulin release. More recently, in the framework of a grant from the European Community 7 Framework Program for a project entitled In Vivo Imaging of Beta Cell by Applied Nanotechnology, we found that 19 fluoro-labeled analogues of D-mannose were indeed suitable tools for the non-invasive imaging of GLUT2 expressing cells, as documented in this article published this year in the Archives of Biochemistry and Biophysics. Moreover, at the present time, we benefit of the quite favorable opportunity to collaborate with Professor Robert Müller and his colleagues from the University of Mons in the perspective of using 13C labeled D-mannose, and in doing so, take advantage of recent quantitative progress in the measurement of 13C labeled compounds. In closing these introductory remarks, I wish again to stress that D-mannose only represents one among at least some other 20 other approaches for imaging of beta cells of the islets of Langerhans, as reviewed in this article, also published in 2012 in Diabetes Research and Clinical Practice, at the invitation of the reviews editor of the latter journal, our colleague Pierre Lefebvre, a past president of this academy. This discours d'accueil facilitates my task in presenting the 2 lecturers who kindly accepted our invitation to this session. First, Professor Kevin Brindle, Professor of Biomedical Magnetic Resonance in the Department of Biochemistry at the University of Cambridge, and a senior group leader in Cancer Research UK, Cambridge Research Institute. Warmly recommended to me by Professor Robert Müller, Professor Brindle has been working since 2006 on metabolic imaging with hyperpolarized 13C labeled cell substrates. And second, Professor Ulf Algren, Professor of Molecular Medicine at the Centre for Molecular Medicine of Ilmenau University in Sweden, and who, in my opinion, provided so far the most elegant imaging of pancreatic islets obtained by the optical projection tomography technology. I am pleased to invite them now to present their lecture. The first lecture by Professor Kevin Brindle is entitled Imaging Tumor Metabolism, Watching Tumor Gasp and Die with MRI. Please.